Hello everyone, my name is Thomas Howard and my fellow presenter is David Hushangi. Today in this 10-minute recap we'll be covering the gene library and quantitative reverse transcription PCR. I'll start out this presentation by covering the gene library, and I'll cover it as the purpose, construction, screening, and components of the library. So the purpose of the library is to create a convenient way to store the entire genome of an organism as clone fragments. This is convenient because it allows scientists to immediately find a gene of interest and begin working with it, rather than having the scientist go sequence the entire genome and find the gene of interest, then use PCR or some method to create copies of the gene. They can just immediately start working with it. And we also have the added benefit of having the entire genome stored as clones and have already been sequenced. There are two different types of gene libraries. We have the genomic library and the cDNA library. The genomic library represents the entire genome of the organism. Meanwhile, the cDNA library represents the cloned mRNA that is turned into cDNA with the help of reverse transcriptase enzymes. The cDNA library will contain varying amounts of gene clones because it is all based upon what genes and how much of these genes the organism is expressing at a certain time. Meanwhile, the genomic library is much larger than the cDNA library, as you probably guessed, because the genomic library contains all the introns and exons of the organism's genome. And so moving on to the construction of the gene library, it is split up into about 5 steps. The first step would be to use a restriction enzyme to cut apart the genomic DNA. It cuts it apart into large fragments and generates sticky ends. During the time that we are cutting apart the genomic DNA, we also are using another set of restriction enzymes to cut apart the bacterial plasmid DNA and also generating sticky ends. The idea is that these sticky ends will have complementary DNA sequences that will be able to bind together and create a recombinant DNA. So step 2, we would essentially mix the genomic fragments and our plasmid DNA, allow them to bind together these complementary ends and use an enzyme such as ligase to glue them together, with our end product being a recombinant DNA plasmid. This plasmid is then inserted into a vector such as a bacteria, and then this bacteria is plated and allowed to grow, and that's where we will get our many clones of our gene of interest through these cloned bacteria. After the plating and multiplication of the bacteria is complete, we would just have to screen the purify our product. So essentially, get the bacteria, take out the DNA, and we would have to screen for our gene of choice. So the construction of the cDNA library is very similar to the construction of the genomic library as the steps were mentioned before. The only real difference is that before we even begin the first step of using restriction enzymes, we have to convert the mRNA into cDNA, and we would do this using reverse transcriptase enzymes. After that, we could just proceed with the same steps as previously mentioned. So going into the screening of the gene library, essentially now that we have all of these products containing our genes of interest, if we have multiple genes of interest, then we're going to have many clones and many genes to sort through. So we have a primary screening and a secondary screening process. As Dr. Wu had mentioned in the previous lecture, after we get all these clones, we may have 100 or more plates to go through. So one method that he had described was for primary screening was that we could go ahead and get 8 to 10 plates and pool all the DNA together, run a PCR, and these 8 to 10 plates would be called the master plates. So we would be running the PCR on these master plates until we found the master plate with our gene of interest. After that plate was found, we could move on into the secondary screening, which is similar to the primary screening, but rather than going, you know, plate by plate or master plate by master plates, we would go row by row or column by column in the master plate to finally find the row or column that contains our gene of interest. We would also be examining the products using PCR, and then once the row was found or the column was found, we could eventually go down and find the actual well with our gene of interest. This method is much more efficient than going well by well and running PCR on each well, because after all, there are many plates that we have to go through. So this kind of allows us to have a method of elimination, and it kind of creates a funnel to allow us to find our gene of interest. So the final section is the components of the gene library. And one of the most important components in even allowing us to construct the gene library is having an antibiotic resistant gene in our plasmid. When these bacteria are plated, they are treated with a antibiotic so that any bacteria that doesn't belong there will die, and theoretically only the bacteria containing our recombinant DNA plasmid will survive that. So this antibiotic resistant gene in the plasmid is extremely important to even building up a library and getting the results that we wish. The second very important portion of it is having a marker gene with multiple cloning sites. Because our gene of interest, we don't get to really decide which base sequences it has. We have to be somewhat flexible on the cloning sites, allowing the restriction enzymes to cut into multiple areas so that the sticky regions of the plasmid and our chromosomal DNA the fragment will be complementary to each other, and they can be able to be ligated. So the marker gene is also extremely important, or marker gene with multiple cloning sites is extremely important to the construction of the gene library. Next, we have the cloning vectors. They can be bacterial or a bacteriophage. The first one is a lambda-derived. That is, lambda is a bacteriophage, and there are different methods when using the lambda-derived one. We have the replacement, which can hold 23 kilobase inserts, and the insertion from cDNA, which holds 5 kilobase inserts inserted into the CI gene. Next, we have the cosmid vectors. It is from the protein of a virus, or the protein code of a virus, and what it does is it nicks a sealed ligase and is able to work using a lytic or lysogenic pathway. We can insert part of the COS gene into the plasmid to increase the capacity of the plasmid to hold larger DNA fragments. Next is the back vector, and those are used to clone large fragments up to 300 kilobases or more. Then we have the bacteriophage P1 and P1 artificial chromosomes, also called PACs. Those are used in circular plasmids up to 122 kilobases or kilobase fragments. Next is a YAC, the yeast artificial chromosome cloning vector, and it allows from 0.2 to 2.0 megabases, which is quite large. This is basically propagated as a linear chromosome, and the mitotic machinery kind of propagates its products throughout the cell, and it becomes part of the vector's genome. But thank you all for listening, and I'll go ahead and pass this presentation on to my partner, David.